The tale of the early Americans is not just one of migration across different landscapes, it's also one of living legacies etched in our DNA, as this ancient genetic inheritance serves as a reminder. Some of the minuscule archaic DNA fragments that were left in the genomes of the earliest Americans by ancient interbreeding between Denisovans, Neanderthals, and ancestral modern humans still exist in Indigenous and Latin American groups today. After diverging from modern Homo sapiens and from each other hundreds of thousands of years ago, Neanderthals and Denisovans eventually mixed with ancestral Eurasian modern humans at various points in time and location. Introgressed Denisovan and Neanderthal fragments were introduced into the original populations of the Americas as a result of these hybridization events, which took place both before and during the times when some descendant modern human groups crossed Siberia and entered Beringia. Indigenous American and some Latin American genomes have been found to contain a Denisovan-derived variant at the MUC19 gene, which affects mucin proteins involved in mucus and saliva barriers. There is evidence that this archaic variant entered modern humans through a complex history of Denisovan and Neanderthal gene flow, and it may have assisted early Americans in adapting to new diets or pathogens. How ancestral Native American gene pools were reached by introgression. Direct interbreeding. Through direct mixing events, Neanderthals and Denisovans each contributed DNA to regional populations of Homo sapiens in Eurasia. Secondary transmission. A layered introgression history that later followed migrations into the Americas is created when certain Denisovan genes emerge in contemporary people after passing through Neanderthal lineages or mixed ancestral populations. Significance in biology and adaptation. Since they provided selective advantages in new environments encountered during the Americas' colonization, archaic-derived DNA fragments, which are typically pretty brief, can actually affect immune response, mucosal protection, and other physiological traits. In at least some cases, these archaic variants appear to have been retained. The date, geographic locations, and functional effects of these introgression episodes are continually being refined by genomic investigations. And interpretations are, tentative and still being investigated because of complex demographic histories, restricted archaic reference genomes, and honestly, gaps in ancient DNA collection. The discovery of the DNA Oreo really changed how we think about our ancient genetic heritage. A study on the gene MUC19, which helps in the production of protective proteins in the digestive and respiratory systems, was published in the journal Science in August 2025. Researchers found that contemporary Mexican communities of Indigenous American heritage have a high frequency of a Denisovan version of the MUC19 gene. This gene variation was found in indigenous groups prior to European contact, according to analysis of ancient DNA from archaeological sites throughout the Americas. Subsequent analysis showed that the Denisovan MUC19 variation is surrounded by Neanderthal DNA in contemporary humans with Indigenous American ancestry. Scientists actually dubbed this peculiar pattern a DNA Oreo, with Neanderthal DNA on either side and the Denisovan version right in the center. According to the research, the MUC19 mutation was originally handed on to modern humans by Neanderthals through interbreeding with Denisovans. Interbreeding between Denisovans and Neanderthals is, one particular instance of the ancient interbreeding episodes that influenced the human genome. And the DNA Oreo is a great example of this. Neanderthal Denisovan interbreeding happened prior to transferring their genes to modern humans, as shown by genetic evidence. As for interbreeding between Neanderthals and modern humans, all modern non-Africans' ancestors interbred with Neanderthals in Eurasia, which is why some contemporary humans have a small amount of Neanderthal DNA. Denisovan human interbreeding also took place. Denisovans, who were mainly active in Asia, interbred with the ancestors of some modern humans, including those in Oceania, Asia, and the Americas. A survival boost. Researchers think that modern humans have adaptive benefits because of this ancient DNA. It's possible that the Denisovan MUC19 gene assisted early Americans in adjusting to new surroundings and fending off strange infections. Long before they migrated to the Americas, Native Americans' ancestors inherited this genetic heritage. The origins are in Eurasia, as modern humans spread out of Africa, not in the Americas. They interbred with Neanderthals and Denisovans in Eurasia. The pioneer's legacy is clear. Neanderthal and Denisovan DNA were inherited by the first peoples to cross the Bering Strait into the Americas thousands of years ago. Over thousands of years, the Denisovan MUC19 variant remained very prevalent in indigenous groups, indicating that it was helpful for adjusting to the new environment. The DNA of Denisovans and Neanderthals still courses through the genomes of indigenous Americans, a living archive of encounters that helped shape survival in the New World.